Hello, everyone. My name is Peter Brodin. I'm a pediatrician, consultant pediatric immunology at Karolinska University Hospital and the Karolinska Institute in Stockholm, Sweden. I am really happy to be with you online to present some of our recent work on the shaping of human immune systems by environmental influences early in life. I want to thank the organizers for inviting me, and I'm sad not to be with you in Rotterdam. I have a conflict of interest slide showing some collaborations or companies that I collaborate with or have been a part of as advisor or founder. So the work in my lab revolves around this concept of systems immunology. And to us, this means an analysis of the immune system, not focusing on individual components, but rather how those components or cell types particularly depend upon each other, regulate each other, and collectively give rise to immune responses to infections or cause disease, as in the form of immune-mediated disorders. So this type of kind of immunological analysis have been enabled by rapid and impressive technological advances made by many groups and companies over the last 10, 15 years. These involve highly detailed analyses of individual cells, such as protein expression using cytometry type analyses, single cell RNA sequencing. It also involves plasma protein analyses, which are now readily done at the scale of hundreds of proteins using small volumes of blood or plasma, as well as, of course, the entire sequences of genes, mRNA transcripts, epigenetic traits, and so on. So in our lab, we try to take advantage of longitudinal monitoring of patients by sequentially sampling patients. And the reason for this is because we believe that this gives us added power to detect patterns of importance, to understand specific diseases or development, which is what I'm going to talk about today, of the immune system. And this is particularly important when you're looking at human patients, given the fact that humans are incredibly diverse as a result of genetic and environmental influences and differences. So today we're going to talk about the development of the immune system. I don't know why it skipped, but anyway, and the development of the immune system is important because of the fact that we've seen a dramatic shift in recent years of disease incidence in the developed part of the world and now increasingly in developing countries as well, which has been a dramatic shift from infectious disease burden that is coming down dramatically, but paralleled with an increase in immune-mediated disorders. And much of this has been traced to developmental perturbations early in life, where the immune system seems to be shaped by environmental factors, and changes to those environmental factors, such as antibiotic use, such as mode of delivery differences, such as infections perhaps, seems to determine the trajectory towards health or disease even much later in life. And this process early in life is what we are keen to understand better. And that's the work I'm going to focus on today. So when a newborn child comes out into the world, it cannot be fully prepared for the environment in which it will reside. The immune system, which is our sensory system for environmental factors, must adapt to the world to which the child is born. The environmental factors, the microbes that colonize the child, the pollutants in the air, the particles around the child, etc., the food components that are being ingested and so on. So all of these things must be sensed by the immune system, and the immune system somehow must know how to either tolerate or reject those different components. And that process is probably very important in determining our immune system even later in life. So we've set out to develop a cohort of newborn children that we follow over the course of the first weeks, months, and even years of life by sequential blood samples. And we analyze these blood samples with respect to proteins in the plasma, cells by mass cytometry, as well as fecal samples that we characterize with respect to their metagenomes by sequencing, shotgun sequencing. And by doing this, we try to just follow how these different children, how they differ from one another, how they differ over time, and in relation to environmental exposure, such as mode of delivery, breast milk or not, infections, antibiotics, vaccinations, and so on. So this is obviously a very large-scale project that currently involves over 300 children, followed for up to 4 years. And so I'm going to tell you a couple of things that we found in this cohort recently. So we published the first analysis of this children in this cohort, the first 100 children, 50 preterm born before week 30 and 50 term children. We also analyzed their parents and their immune system. We worked hard to be able to extract as much data as possible from small blood volumes available from especially the preterm children. And we are now able to confidently assess all of the blood immune cell populations, including neutrophils. So this is a whole blood analysis by mass cytometry, as well as 267 plasma proteins, and we can include a few more even using O-link assays. So in the first analysis, here's a summary because this is published work. In this summary scheme, we use an unsupervised analysis method called topological data analysis to organize the samples based only on the data that we've measured. And what we find is that cord blood is an outlier tissue, very different from the postnatal immune system. And we find that the immune system follows a trajectory over time with dramatic changes during the first 3 months of life. But those dramatic changes are very similar between preterm and term children, leading them to converge on a trajectory that is shared, such that around 3 months of life, these children are now inseparable with respect to their immune system. So this was interesting and led us to ask a lot of additional questions. But what we do believe is that this represents a physiological adaptation from the immune system to extrauterine life. We know that this is the greatest change in environmental exposure anytime during our lives, and the immune system must adapt to that. But what we've done since is we've tried to characterize in more detail and with more children how these different changes occurs. And what we've found is that there are sequential waves of immune cell reactivities or reactions that I want to tell you a little bit about. So here I'm showing you mass cytometry data with individual immune cell populations on the left, and then also O-link data with cytokine measurements on the right. And what we see is when we bin the samples based on the day of life at which they were taken, we see that there's an initial dramatic expansion of classical monocytes in the blood, which is paralleled with an interferon gamma increase in circulating plasma. Following this initial response is an expansion of memory phenotype regulatory T cells, perhaps as a response to the initial innate activation. These Tregs expand in the blood. What they have seen is yet to be determined, but it still seems like a mechanism to limit the inflammatory initial response. Following the memory Tregs from about 1 month onwards, we see an expansion of gut homing cells such as gamma delta T cells producing IL-17, these CD161 positive cells, as well as a small but significant increase in mate T cells, all presumably going to mucosal surfaces. And finally, from about 2 months onwards, 60 days at least, we see an expansion of activated CD8 T cells and these effector memory CD4 T cells, which I'll tell you a little bit about in a minute. So one particularly interesting cell type to us is cells that are involved in mucosal surfaces. And the reason for that is because we know that at the same time frame, children are also being colonized in their intestine, on their skin, and everywhere, in lungs, of microbes. And from work based in Janneke Sandsum's lab, also in the Netherlands, we have learned that memory CD4 T cells of this phenotype expressing CD38 but not 62L, those cells are not so abundant in the blood of adults, about 4 or 5%, but they are dominating in the intestine. So from this biopsy of a healthy individual, all of the T cells are of this phenotype. And what we see in newborns is that these cells are much more abundant already at birth, 30% or so, and this increases over time during the first months of life to up to 65%. And when we sort these cells out and we compare them to the total memory CD4 T cell compartment, we see that these cells are enriched for interferon responses, both gamma and alpha, which we think is intriguing and speaks a little bit to their possible function in the mucosa. And when looking carefully at these cells, we see that they seem to have a regulatory role. They're producing cytokines such as IL-15. They're producing CSF1, which is known to influence monocytes, of course, in the tissue perhaps. And so we believe that these might have an immune regulatory role. We're also looking at the the microbiome as it's colonizing these children. And we assume that most of the colonization, if not all of it, happens at the time of birth and after birth. We don't believe that there is much of a microbiome present before birth, and that's based on the literature that we see as we see it. We do see that there are dramatic changes in the microbiome, the gut microbiome, as measured by fecal samples. For example, we see large amounts of skin bacteria in the C-section delivered babies. That's well known from other people's work. There's high abundance of Bacteroides in the vaginally delivered initially, which is also known from before. The most dramatic change over time, early after birth, is expansion of bifidobacteria. And mirroring this expansion of bifidobacteria, which in our case is seen very clearly at 100 days of life, because we lack samples early here, we see also a dramatic increase in short-chain fatty acids during this time, which is well known to be produced, for example, by bifidobacteria. And so we think this speaks to this change in the microbiome with an increase in bifido. But the children do not all behave the same way. And so basically we can summarize 3 different patterns during the early life colonization of the gut. And we can use bifidobacteria as a surrogate marker for 3 different types of microbiomes. Obviously a simplification and generalized pattern, but still I think useful for further analysis. And we see that most of the children increase with respect to bifidobacteria, but a few children are completely devoid of bifidobacteria throughout this time. And we decided to compare their immune systems. And when we do this, we focus in on the samples collected around 3 months of life, sort of at the end of this phase here. And what we see is that the children that have low abundance or no bifidobacteria have much higher levels of IL-6 and also oncostatin M, which is an interesting cytokine mediating inflammation in the intestine, for example, in IBD patients, as shown by Fiona Powery's group at Oxford. at Oxford. We also see higher abundance of IL-17 and CXCL11 in these children with low bifido. With respect to cell populations in the circulation, these cells, these patients or children have much higher abundance of activated CD8 T cells, as well as pro-inflammatory monocytes and neutrophils in circulation, while the patients with increased amounts of bifidobacteria have more of class-switched memory B cells and plasmablasts. We're also, I mentioned in the beginning that we work along this systems immunology type paradigm. And what that means to us is that we're interested in how cells cooperate and give rise to behaviors in the immune system. And one way to look at that, which is a rather simple way, is to look at cell-cell interactions or cell-cell correlations in this case. And what we see then is that there are some correlations, such as the very strong correlation between 3 types of naive T cells that is shared among bifido high and bifido low children, presumably reflecting the function of the thymus. But there are other relationships that are completely different. For example, we think that this more healthy immune microbiome interaction leads to, which is seen in the high bifido children, that leads to a strong inverse correlation between memory Tregs and activated immune cells. And this relationship is completely lost in the children without high bifido. So what this suggests is that perhaps there is a updated model in which the first 100 days seems to be critical, and where bifidobacteria has a beneficial effect by reducing inflammation and allowing for the formation of a balanced immune cell network. And so we teamed up with a company called Evolve Biosystems, which is out of San Diego. And they had performed an interesting trial, which was to provide as a supplement bifidobacterium, specifically the subspecies infantis, through a product they call EVC001. And they gave this to full-term breastfed infants and compared those to children receiving no such a supplement. And what they found when looking at fecal cytokines was that they were comparable at before intervention, but at day 60 after the intervention, the children that had been given the infantis bifidobacteria had much lower levels of IL-17 and also lower levels of IL-4, which are, Th2 skewing cytokine, while these children who received bifido had much higher abundance of interferon beta, which is known to induce regulatory T cells, for example. So this was intriguing, and we tried to take this a little bit further, and we set up cultures of naive T cells in vitro, where we performed sort of typical polarization experiments using sorted naive CD4 T cells. When you culture those in specific cytokine cocktails, you can skew those into these different lineages, Th1 to 17, and induce Tregs. And we did so in vitro, and then we provided either fecal water from these children that I previously shown you from the trial with high abundance of bifidobacteria infantis, or children without such bifidobacteria infantis. And we used a single-cell RNA sequencing approach, which is a targeted approach, combining both the analysis of antibodies, proteins by antibodies, as well as mRNA. And what we see here in this slide, these are two-dimensional projections of the T cells in culture. And the first thing that's pretty notable is that these cultures differ significantly with respect to Th0. So these are cells cultured without polarizing cytokines and only get TCR cross-linking. What we see then is that in the presence of low fecal water from bifido low children, the cells basically assume a Th2 type profile, as you can see here, while children that have been that have been cultured with fecal water from bifido high children assume a Th1 type skewing here. So the somehow metabolites in the fecal water is skewing T cell polarization away from a Th2 type phenotype to a Th1. And this is also seen here where the bifido-high cultures have high abundance of IL-2, RA, granzymes, TNF, while the bifido-low have these genes upregulated, such as IL-23 receptor, for example, which is an interesting receptor mediating, for example, inflammatory responses in the gut. So with this, I would like to thank this phenomenal group of people that I'm working with at the Karolinska Institute, my colleagues in the hospital that work in the neonatology center and help us with sample collection, as well as the collaborators at Evolve Bioscience. Thank you very much.